You mean this lecture?
No, the previous test. The previous test.
I mean the oral exam.
Yes.
It was very bad. Very, very bad. You were all, I think you all failed. You all failed. So, yeah. It was not good at all.
So what do we need?
So that, no, I'm just joking. It's just what I told him. It was, I'm very happy because you all did very, very well. I think you all have A or A plus. I'm really, really happy because I had fun discussing with you because you had all kind of good ideas, and not only you understood what I said during the lecture, but yeah, I'm very, very happy. There is nothing to worry about. Very, very, very. It's stressful, right, when you come and speak, isn't it? It's very stressful, but you should not worry. It's yeah. I was very happy.
I realized I have to practice my speaking English.
Yes, this is one comment. Is that you have very good idea, but sometimes the level of English prevents you to. But that's the case of many people. Prevents you to clearly say what you have.
Sometimes it's hard to explain in what ways because sometimes I didn't have a clear idea to express. So it's not all. It's not the problem. Is not all about English. It's about how to speak, how to express ideas.
Yes, that's true. It's not only a language thing. It's organization in the brain, and yeah, that's true. But you should not worry. It comes with practice. So. It's already the beginning, right? It's 2:45, right? This lecture. Okay, let's start. All right. So, maybe let's wait 5 minutes more. Just finish my drink. Somebody else may come in 5 minutes.
What was your question?
What is temperature compensation and why is it important? And I have given experiment a piece of paper and mutate rat and shorter sleep cycle rat have produced, and
Rats that have some mutations in some kind of in some protein like clock or lemon.
Protein is not specified, but some mutation and.
Okay, that's all right. So, I have not planned anything for today. No lecture, no paper, nothing. But since we are only 2 people, well, we are 3 actually. I mean, 2 students. I'll just talk to you about what I'm doing, about my research. But you cannot speak about it. Okay, you cannot speak about it.
Confidential.
It's confidential. Okay. In the beginning, most of the thing is already published, so you should not worry. But once something is confidential, I will tell you. Okay. So you know from previous lectures that scientists think that the clock, the circadian clock in mammals, is a transcription translation feedback loop, okay, of clock genes. And although this is known for a long time, more recently some researchers have said, by the way, I think there is a possibility that in mammals there is an oscillator, there is a pacemaker that has nothing to do with the genes. It's something that is more biochemical. Okay? And this is very exciting because maybe it's true that in our cell, the whole biochemistry of the cell is a clock. So, what I want to ask you is, if you are interested in this, if you want to know what could be the clock in the in biochemistry of the cells, what kind of experiment would you like to do? You want to know what is the pacemaker in the biochemistry of the cell. What would you do? What kind of experiment would you do to get insight into, this might be interesting, this might be the clock, or this might be linked to the biochemical clock? What kind of experiment would you do, please?
First of all, make a list of all the biochemicals that the cells that are oscillating in the cells.
Very nice. Excellent. This is exactly what I did. This is exactly what I did. And so not only we made a list of all the things that are in the cells, but we quantified all of them. We quantified not all the genes, but ATP, ADP, CTP, amino acid, just everything that is in the cell. We quantified everything. And it's a lot of stuff. There is a lot of data. Maybe it's about, of course, we cannot detect all because some of them are very low concentration, so we cannot detect all, but we detected maybe 200 or 300 biochemicals. And what we did is that we checked the amount of these biochemicals in the liver every 4 hours for 24 hours. So we know if there are rhythms in biochemicals or not. Okay, do you understand this? Do you understand? Yeah?
These many substances, or whether this is not by transcription translation feedback and this is not by this, you cannot understand.
Exactly. Although you will be able to know whether they are rhythmic or not, you will not really be able to know the rhythm in this biochemical. Does it come because of gene expression rhythmically, or does it come because of biochemical interaction? You don't know, that's true. But what you can do is using a mouse that has no clock. You can use a mouse that has no transcription translation feedback loop. For example, what could you use? What kind of mouse could you use that has no biological clock? Completely broken transcription translation feedback loop. What kind of mouse could you use?
What kind of mouse?
Yeah, what genetically modified mouse could you use that has no clock?
I first thought you just use red blood cells.
Yeah, but red blood cells is very difficult, and the amount of metabolites that you can get from red blood cells is very low. So you need a lot of red blood cells. You probably need to take all the blood from one patient. So this is good, but it is not so good. And then you need to do every hours, and then you want to quantify everything. So it's just not one single protein. You want to quantify everything, so you need a lot of material. And using the liver, it's very good because the amount of metabolites is very high.
May I ask their SCN removed?
Mouse with DSCN removed, it's a good idea. However, as we discussed during the exam, the mouse with DSCN removed may still have rhythm in their liver.
What remove?
SCN.
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 Suprachiasmatic nucleus.
Ah.
May I say they have mutation in the clock protein, like the clock protein?
Exactly. So this is exactly what we use. We use the mouse. Of course, we compare. We compare the mouse, a normal mouse, and we compare this mouse with the mouse who has no more BMAL1. And the mice that have no more BMAL1, they are completely arrhythmic. No more rhythms at all. Okay.
They can live.
They can live, yes. But they are they are not healthy. They are quite small, and some mice tend to die quite quickly. So they are not very healthy, but they don't all die. But you can definitely see that. But you don't really know what happens. Are they unhealthy because they don't have clock? Or are they unhealthy because they don't have BMAL1? This is slightly different. You understand what is the difference? BMAL1 is important for the clock, but BMAL1 may be also important for other things. So you don't know if the BMAL1 knockout mice have some disease because they lack the clock or because they lack BMAL1. It's a little bit. Okay, so we quantified many things in the liver of these mice, and now you can see, for example, a picture of these things here. So it's a lot of them, okay, a lot of them, and this data only show a few of them, the ones that were significant. And we checked, this is the data that show only 2 time points, CT4 and CT16 in wild type, in a normal mouse, and CT4 BMAL1KO and CT16 BMAL1KO. And then we checked how are the amount of all these metabolites affected by time and by the knockout. And then we analyzed statistically, and we asked, what is the metabolic pathway? You understand? What is the metabolic pathway that is the most affected by time and by the lack of clock? Okay. So we wanted to find something that has big rhythms, but something that is not so different between BMAL1 and wild type. That will still be rhythmic, even though they have no BMAL1. Okay? So we did this analysis. Tell us what is really time dependent. And then the analysis we got is this one here. This one, as you can see, can you read? Yes? So this is when it's red and when you have a big bar, it means that it's very significant. Okay? These are all the pathways that we have identified. So we have all our metabolites, and of course they are grouped by different pathways. For example, you have here insulin signaling, gluconeogenesis, glycolysis, purine metabolism, like a nucleotide metabolism. So all these metabolites, they are part of this big family of metabolites. And what this analysis told us is that, okay, interestingly, protein synthesis was very significant. But that is...
Significant in time dependent?
Yeah, that is because we got many amino acids. We got many amino acids with completely different levels. So many amino acids, this computer will see, okay, you have many amino acids that have big difference between CT4 and CT16. So protein synthesis is important. But for us, it only means that we have a lot of amino acids. Okay. The one underneath was very interesting. Why? Because we saw methionine metabolism. Okay, this is still amino acid, so it's this methionine metabolism is a little bit the same as proteins biosynthesis, but it's a bit more specific. But then methionine metabolism, glycine, serine, threonine metabolism, that's interesting. And then I jump one, glutathione metabolism, betaine metabolism. So that's very interesting because what we noticed is that the second one, the third one, the fifth one, the sixth one, and even cysteine, they are all linked by one very specific metabolic pathway. They are all linked by one thing, and the thing that they are linked by is something called the methyl cycle. And this is, I show you the synthesis of all these metabolites. So this is this thing. So this one here is the methyl cycle. Why? Okay, so that's all right. So this is the methyl cycle. And then you have methionine, SAM, SAH. So all these is called the methyl cycle. Okay. And then this regulates betaine metabolism, which was also something with, as you can see, very strong rhythm here, always. Boyon, listen the other way around, same rhythm here. This methyl cycle also regulates cysteine metabolism. And cysteine metabolism, cis is cysteine, right? Cysteine metabolism, and also regulate, this is glutathione metabolism. So they are all linked by this very simple methyl cycle. Methyl cycle, I explained to you why. You see, it's 4 metabolites. Methionine, that is used to make S-adenosyl methionine, SAM. This is okay, now we can read. So this is methionine. This is SAM, S-adenosyl methionine. And this is SAH. So this is after using SAM, you make SAH. And after using SAH, you make homocysteine. We did not detect homocysteine because the amount of homocysteine is very low. So we did not detect this one. But then homocysteine is used by using betaine and regenerating methionine. Okay, this is a cycle. Methionine is used to make SAM, and then it's regenerated into methionine, and again, and again, and again. And something that we were kind of interested is that when you actually check, I will explain to you why, but when we make the ratio of SAM over SAH, so we divide the data of SAM by SAH, this is what we get. So this is the ratio between the two, pion. And this is in wild-type mouse. You can see it increased very strongly between CT4 and CT0 and CT4. And then it goes down, and then up during the night, and it goes high again, like this. But then in BMAL1 knockout mice, we had the same difference between CT4 and CT16. The variations in SAM and SAH was very similar. Even though the mice do not have a clock, they are in constant darkness, so even their food intake is arrhythmic, but still the difference between CT4 and CT16 was the same. So somehow the rhythm of this SAM-SAH ratio was very similar. And if you take SAH here, CT4 is low, CT16 is high, but also in BMAL1 knockout mice, this is red here, between CT4 and CT16, there is still an increase. It's a little bit lower, but we still have an increase. So we were very interested by this, because the data told us that this pathway, the whole thing, is enriched with metabolites with time-dependent differences. And not only that, but the variations of most of these kind of remain in BMAL1 knockout mice. Okay. Have you heard about the methyl cycle before?
No.
S-adenosylmethionine. Okay, so the methyl cycle makes SAM, and SAM is, it's not a name, it's S-adenosylmethionine. So of course this means it's made of methionine, right? And it's made of adenosine. S-adenosylmethionine, it's a specific molecule that is also known as methyl donor. Methyl donor means it can give a methyl. Now, methyl, you should know what is a methyl. Methylene, right? It means that somewhere in the SAM molecule there is CH3. And this CH3, this methyl, can be given to other molecules by enzymatic reaction. For example, what? What do you know in the cell is methylated? If you think about all the components in the cell, what is methylated?
DNA.
DNA, very good, is methylated. What else? Protein is methylated, especially histone protein are methylated. But not only histone protein, but also proteins in the cytoplasm are methylated. What else? Actually, just about everything. Okay, DNA is methylated, protein is methylated, fatty acids are methylated. Metabolism of fatty acid is important. Methylation is important for the metabolism of fatty acid. RNA is methylated. Ribosomal RNA, transfer RNA, messenger RNA. All RNAs are methylated. Everything in the cell is methylated. And methylation of these macromolecules is very important for their function. For example, you said DNA, you said protein. So if we focus on DNA and histone methylation, what is the effect of methylation? Do you know what is the effect if you methylate DNA or if you methylate histone? What is the effect it has?
DNA will be condensed.
Yes. DNA will be condensed and you stop gene expression. What about histone methylation? Histone is the same. But depending where you methylate the histone, it can condense or it can promote methylation, depending which residue in the tail of the histone is methylated. So methylation is very powerful. It's something that can regulate the function and the activity, the stability of things. Okay. This is very interesting. And what we have here is that this whole pathway that makes SAM is very strongly rhythmic. even apparently in mice that do not have biological clock. And why is this important? Why did we make SAH is the same as adenosyl, but it's not methionine, it's S-adenosyl homocysteine. And SAH is made because when you give the methyl of SAM to different target macromolecules, SAM, of course, is degraded and is transformed into SAH. Okay? So SAH is the product of methylation. And SAM is used by enzymes that methylate macromolecules, but SAH is an inhibitor of methylation. So SAH has allosteric inhibitory effect on methylation reaction. So in this case, what means the ratio of SAM over SAH? What does it, mathematically, what does it represent? Well, mathematically, it's just the ratio. You divide the amount of SAM by the amount of SAH. But biologically, what does it mean?
Go into the equilibrium. It went into the equilibrium when the concentration of SAM does not change.
Yeah, so it has to do, that's right, it has to do with the concentration of SAM and SAH. Imagine two cases. There is a lot of SAM and there is not a lot of SAH. So then the ratio becomes high. So if there is a lot of SAM and a lot of SAH, the ratio is very high. So there is a lot of SAM and there is little of SAH, the ratio is very high. But the other case, there is low amount of SAM and there is a high amount of SAH, then the ratio is very low. So what do you think will happen? And you can see the ratio changes. And during the day it is high, during the night it is low. This is the ratio, okay? So what will this ratio mean? In the cell, during the day, the ratio is high. During the night, the ratio is low. What do you think will happen if SAH is an inhibitor of methylation?
It will fluctuate over time.
What will fluctuate?
The concentration of both.
Okay, so this is, the fluctuation will change. We can see it changes. And because the fluctuation of SAH and SAM fluctuates in empty phase, you can see SAM is high during the morning, but SAH is low during the morning, okay, here. So of course when you divide, you have fluctuation. But what this fluctuation means? What consequence will this fluctuation will have on methylation reactions in the cell? DNA methylation, RNA methylation, protein methylation, histone methylation. These changes, these fluctuations in the ratio, what effects will it have on the methylation of various macromolecules in the cell?
During the day, methylation reaction may occur frequently.
Yes, exactly. This is exactly what happens. So this ratio, the ratio SAM-SAH is known as the methylation potential. And methylation potential means that when the ratio is high, then methylation reaction will happen very quickly. And many methylation reactions will happen. But when the methylation is low, then the methylation potential will be very low, and methylation reaction will not occur. Okay, so this means maybe during the day there will be a lot of methylation reactions, methylation of DNA, methylation of RNA, protein, but during the night they will be very low. So which means that the stability, the activity of DNA, RNA, protein may be regulated by this variations in methylation potential. So imagine if this is rhythmic and drives rhythm of methylation potential. So maybe you could have then a mechanism by which this regulates gene expression of many things as well. Because it is changing and it regulates histone, it regulates RNA methylation, it regulates DNA methylation. So maybe what we have here, it's a kind of metabolic oscillator that can regulate many different things during the cell. Okay? So what we thought is that maybe this methyl cycle is maybe a kind of oscillator in the clock, a metabolic clock that can then regulate gene expression by regulating protein, RNA, and DNA. So that was our theory at that time. We see all these rhythms, methylation potential changes. That's kind of interesting. So, let's say we want to test our theory. And we think, okay, since the current model of this, biochemical oscillator and TTFL, so I can write this one here. You have the biochemical oscillator, and you have the TTFL, the normal TTFL of the clock. It oscillates. And the current model of this is that the biochemical oscillator regulates the TTFL, they reinforce each other, but the TTFL also kind of interacts. Okay? So the two are kind of coupled. And what we have here maybe is that the methyl cycle may be a biochemical oscillator that regulates via methylation of many things the transcription translation feedback. So that was our idea, and that maybe some SAH is maybe one of these biochemical oscillators. Okay, we want to test this hypothesis. What could you do to see, okay, is this methylation potential important at all for the clock? That's the first thing we want to check. Is it regulating the clock? What could you do? You want to test whether this is true. Whether this some SAH really kind of regulates the normal TTFL. What could you do? What will be the next experiment to test?
Make this value constant.
Yes! Very interesting. You make the ratio, you make the methylation completely constant. Yeah, that's one of them. How could you do that? By the way, that's very complex because all these, of course, this here, there are many different enzymes. There are different enzymes for each. So there is an enzyme that methylates DNA. Well, there are many enzymes that methylate DNA. There are many enzymes that methylate RNA. There are many enzymes that methylate protein. So this reaction is really non-specific. Many things makes SAH. But here there is one enzyme that removes SAH and transforms it into homocysteine. And then there is maybe one, depending on the tissue. Actually, there are two enzymes, but there is only one enzyme expressed in each tissue. So there is one, let's say there is one enzyme that then uses homocysteine to make methionine. Okay? And methionine from SAM, let's say for the sake of argument, there is also one enzyme. So these enzymes are all different. And this one, many. It's a very complex pathway. And you want to know whether these oscillations in methyl cycle is important for the TTFN. Your idea is very good. You make it constant. How could you kind of make it constant? Or how can you change its level?
Ratios to fluctuate in person, but because I don't understand why don't they get into the case that into the situation that SAM the EMF of SAH is right and is right, so that it will not change the EMF of SAM. So
once more.
I don't understand because we have two sources to make SAM. This one source is for no. We have only one source to make SAM, and it is from SAH and the SAM can go into two ways. One is to return back into SAH, and one is to methylate other substances. And for SAH when SAH gets to a certain value, then the reaction, then sorry, I was wrong.
It's okay.
So SAH is constantly transferred to SAM no matter?
No, SAH is not transferred to SAM. SAM, during methylation reaction, SAM is transformed in actually SAH is SAM without its methyl.
Yes.
It gives its methyl to many things, and then it's transferred to SAH. But then the cell has to remove SAH, because I told you SAH is an inhibitor of methylation. So the cell must remove SAH, and to remove it, it transforms into homocysteine. And homocysteine is very bad for health. There is a disease in humans that is called hyperhomocysteinemia, and it makes a lot of heart problems. So this is very bad. So the cell also must remove this, and to remove homocysteine, it will make methionine. And then methionine, again, can be used to make SAM, and so the cycle continues.
And it is also homocysteine can be also changed into glycine.
Yeah, so actually homocysteine, a part of homocysteine goes into methionine, but there is another molecule, right? There is betaine plus homocysteine. Both are transformed into methionine and dimethylglycine. It's kind of big molecules, okay? So they are coupled together, but then they separate and give two other molecules. So how could you sort of make the methylation constant, the methylation potential constant. How could you make it? I should take back. And this is a very common, it's a very common way in biology when you want to sort of investigate some metabolic pathways. You usually do. This is something you usually do. Considering the department we are in, it should be easy to understand. We are in the pharmacology department, so how could you change? If, of course, the SAM-SAH ratio depends on the amount of SAM and the amount of SAH, of course.
Can you make another kind of methane differ?
Another kind of
Methane differ.
Of?
Methane donor.
A methyl donor. Yeah, I mean, you could put a lot of SAM in the cell. Into the culture medium, you are studying cells, and into the culture medium, you could put a lot of SAM. You could do that, but the problem is the cell will use up the SAM. You may be able to affect the clock, but very quickly SAM will be degraded by the pathway. So you must find a way to actually, not permanently, but to affect the pathway for a long time. Because you want to see whether there is an effect on the biological clock. So it has to be for a long time. How could you do?
Take off the SAH.
You can take off all of the SAH. Yes. You could also do that, and then you have a lot of SAM, and so the methylation potential will be always very, 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 very high. That's true. But removing SAH is very difficult.
You could activate the enzyme that helps to transfer SAH to homocysteine and homocysteine.
Yeah. So you activate this. You activate this. That's good. That's very nice. Unfortunately, there are no activators. If you look on the database of chemicals, there are no activators of this enzyme. This enzyme is called adenosyl homocysteine hydrolase. Okay, this enzyme, but there is no activator of this. So this is not good. Mechachika, you are very close. You are very, very close. Very, very close. There is no activator of the enzymes.
No, you put a lot of enzyme.
Putting another enzyme is very difficult because then you must maybe do some sort of transgenic cells, and then you want to be something more simple that you can check almost tomorrow. You are getting farther away. Your previous idea was very close.
If you inhibit the enzyme,
Yes.
the ratio,
There is no activator, but there is an inhibitor.
the ratio will be 0.
Exactly. You have, actually no.
No.
You are using an inhibitor, and it's an inhibitor that prevents the removal of SAH. So SAH will become always high because the cell only has one way to remove SAH, it's this way. So if you stop this, SAH will go high and high and high and high, always high, and it means the methylation will always be inhibited because SAH will always be high. So the ratio will be always very low. Okay? The ratio will be extremely low. And there are many inhibitors of that. Okay, so that's nice. That's very nice. So I can show you the next figure that we sort of did. And maybe this one is bad. Yeah, that's not bad. So what you have here, DAA, it's I will write this one. It's D other adenosine. Okay. So adenosine, it's a nucleotide, just like all these things. Okay. Adenosyl methionine, it's nucleotide and amino acids together. So there's adenosine. It's a molecule that looks like homocysteine. It's not so different from homocysteine, but it's an inhibitor of the reaction. It's an inhibitor of the reaction SAH to homocysteine. So when you put deoxyadenosine into the cell, the methylation potential goes very low. And all the methylations in the cell are inhibited. Do you understand what I'm just saying? Yeah?
DAA is inhibitor of
That reaction.
the reaction.
Yes. So DAA is an inhibitor of the reaction. And then this, you can see here, it's marked. You can forget for now the detrending, but this is luminescence. Luminescence, why luminescence? Because we are using a specific type of cell that is per-to-look. Okay? There are many things. There are many things. So, yeah, I will continue using the board. There was an exam question, I'm not sure who had it, but I think it's the guy here. So, okay, I go back a little bit in one of the previous lectures. You want to measure the expression of clock genes. You know, these clock genes oscillate, right? So if you want to measure the clock, you have to measure oscillations of clock gene. Okay? But how can you measure oscillations of clock genes? You have cells, and what you want to do is to test what is the effect of your drug on the oscillations, on the transcription, translation, feedback loop. But for that, you need to measure either the amount of RNA or either the amount of protein, right? How can you do that? Easily.
Tagging with antibody with fluorescent protein.
Yes, you could do that. You could use antibody that are targeted against, for example, PER2 protein. And then if PER2 protein is high, then the antibody will go into the cell and bind to PER2 protein. And if your antibodies are tagged with GFP, then the cell will be green. But if there is no PER2 or very low, then fluorescence will be low. This is okay, but the problem is, every time you want to measure, you have to kill your cells. Okay? So let's say you want to check your rhythms for 1 week. Every 4 hours, you will need to have many cells. You want to study this like in real time. At any time, you want to take a look at your cells and see. So this is okay, this is nice, but it's not so practical. What else could you do? This gives you a big hint. This is luminescence. Luminescence means the amount of light. To do what? How do you do this luminescence?
To find the target gene to the luminescence.
Yes, exactly. The words you use are not so precise, but this is what you do. So you have a gene, and this is the beginning of the transcription, and this is PER2 gene, for example, in this case. This is PER2 gene, and to simplify things, you imagine that, so this is the mRNA, and then you have the coding sequence, and this will be translated into the PER2 protein. But what you can do is to genetically modify the mouse and put here, just after the coding sequence of PER2, you put luciferase. And when it is translated, it will be translated as one protein that, if you are lucky, will work like PER2, but will also work like luciferase. So this means that you can use these cells who are genetically modified, and you just need to measure the amount of light they make. If they make a lot of light, because luciferase, you know what is luciferase? Do you know luciferase?
No, but it maybe produce light.
Hotaru no tonpakujitsu. Okay, it's a protein of a firefly. So you make these cells, and then the amount of light when it's high, what does it mean? That's a very easy question. So when these cells emit a high amount of light, what does it mean?
Many PER2 protein will.
Yes, many PER2 proteins, yes. When the amount of light is very low, what does it mean? Exactly. So then, by using these cells, you can, just by measuring the amount of light, you can know how much these cells make PER2 protein. And this is the data that you have here. So you can see, look at the black line. The black line is cells that were treated with 0 micromolar of DAA. No DAA at all. Okay? And you can see they are oscillating with a period of about, these cells are a little bit funny, but 25, 26 hours. Okay? And they oscillate very nicely. But then, when you add 100 micromolar of DAA, you can see what happens. And then what you can do, you can see this is 120 hours, a little bit more. And then after that, you wash the medium. You remove the DAA from the medium, and you replace the medium with normal medium. And then suddenly, these cells, they become very similar again. What is happening then? Well, you can see here, if you actually calculate the period of the oscillation, how do you calculate the period? How could you very easily, there are more complicated ways to do, but if you have this data here, how can you calculate the period? Very simply.
Minus the time between 2 weeks.
Yeah, exactly. So you take the time of this peak, and you calculate how much time it takes to reach the second peak. And then you calculate how much time it takes to reach the next, and the next, and then you can calculate the average. And when you do that, you can see that with DAA, the period is about 26.5. In control cells, the period is about 26.5, but with DAA, the period became 29 hours. Extremely long period. But then after washing off DAA, then you can see that the difference between the cells that were treated with control and cells that were treated with DAA, there is almost hardly any difference. So somehow, inhibiting the enzyme that is responsible for the removal of SAH had a very strong effect on the transcription translation feedback. So there is definitely a link between the methylation cycle and the transcription translation feedback. You don't know what it is, but there is definitely a link. Okay.
SAH inhibit every kind of methylation reaction, or it only inhibit?
That's a very excellent question. And it's a question we had during the research. Does SAH inhibit every methylation reaction? What do you think? I told you methylation of DNA, methylation of protein, methylation of many, many things. Many methylases, if you look on the database, there is about 300 proteins, different enzymes that methylate different things. So do you think they will be inhibited all by SAH? What do you think?
I think there is many other methyl donors.
No. There is only one methyl donor. This is the beauty of the system. There is only some. There is only one methylation reaction that requires a different one. Okay? But most, 99.9% of the methylation reaction, they require SAH. Okay? But SAH, I told you, is an allosteric inhibitor of methylation. Do you think all methylations will be inhibited by SAH?
Is the allosteric inhibitor that inhibit every methylation with SAH? Yes.
No. Well, yes, it might. It may inhibit all, but actually, if I tell you that the structure of these proteins, of these enzymes, the enzyme that methylates things, they are called methylase or methyltransferase. Okay? And the structure and amino acid sequence of this methyltransferase is quite different. So then, what do you think? If the structure of this protein is different, if the structure of this methyltransferase is different, do you think that SAH will inhibit with the same power all the methyl? No, you are right. So each methyltransferase will be inhibited or not by SAH. Some methyltransferases are very sensitive to SAH. Some methyltransferases are not at all. It really depends on the structure of these proteins. So what we are having here, it's a result that depends on methyltransferases that are probably highly inhibited by SAH. Okay, but we don't know which one. Because as I told you, there's 300 of them. So we don't know what is the inhibition of all of these 300.
DAA inhibits the cell state M of HAS, so DAA increase the DAA function is only to increase the SAH in the cell.
That's an excellent question as well. So DAA, actually this is a question that when we published the paper, it was asked by the reviewers. And we say, well, you are using DAA, but DAA has also other effects. DAA, as I told you, it's a nucleotide. There's an adenosine. So it's also used by the cell to make ATP for DNA synthesis. So there may be other effects that diazoadenosine has that is not dependent on methylation. Some side effects of DAA. Okay? So this is why I'm going very deeply there. So then we had to find another molecule. We had to find another chemical who could still inhibit the enzyme that hydrolyzes SAH to homocysteine without having any other effects. And there is one. This is called diazaneplanosine. Diazaneplanosine. And the basic structure of diazadenosine is that it's a nucleotide, so it contains a ribose, the sugar of the nucleotides. And this is a carbon, this is a carbon, this is a carbon, and this is also a carbon. And because this is exactly like adenosine, this is why the cell can use this also to make RNA and everything. So it affects many other things than just methylation. However, some very clever chemists, they made a molecule that is just like diazadenosine, but they replace the oxygen by a carbon. It's just exactly the same structure, but this oxygen has been replaced by a carbon. And because of that, this diazaneplanosine only inhibits SAH to homocysteine. It cannot be used as a nucleotide. And we got the same data. So what do you do next? When you inhibit all potentially many methylations, the clock is affected. So what is the next step? And of course, you understand that at this stage it's very general. It's not at all specific. We just inhibit everything. Potentially inhibit everything. But we don't know what we inhibit. We inhibit methylation, but the methylation of what? So what is the next step?
In the catalyst of the cheese that oscillate in ordinary situation and cheese that do not oscillate in DAA culture. Checking which gene oscillate when there is no DAA, but when start to oscillate.
No, it's kind of a nice idea. I didn't do that, but it's a nice idea. We checked many genes. We checked many genes to see, okay, maybe this is only an effect that is dependent on PER2 look. Okay? But if we use other cells, like this is mouse cells, okay? But if we use other cells, do we have the same effect? And I can show you the main thing. So that's what is that? That's the paper we published some years ago. It was insane. I was happy. So that's the data I just showed you, but shown a little bit differently. Maybe a little bit easier to understand. Okay, so this is the same data. Okay, so this is using mouse cells, and then as you can see, in addition to what I showed you previously, we tried different doses of deaza-adenosine, but it works the same with deaza-niflanosine. And then we used 50 macromolar, 100 and 200. And as you can see, the more you put, the longer the period becomes. This is in mouse cells. And we got exactly the same data in human cells. This is human cells that are also genetically modified to express luciferase under the control of BMAL1 protein. So this is BMAL1 LUC, and you can see the same data happens. So the period increases the more DAA we put. And so we checked also many other genes. So this is not luciferase, this is all luciferase. But we also check the real amount of RNA, and this is the amount of RNA of PER2 in normal cells. And as you can see, under DAA, we have the same data. The period is longer. And we checked BMAL1. This is another name for BMAL1. And the same result again happened, longer period. We checked, this is in mouse cells, and in human cells, you can see the period is completely different. And because the period is different, the phase is also completely different. This is human cells, PER2, BMAL1, and this is mouse cells, PER2, BMAL1. So many genes are affected in the same way. So it's a completely global thing. But the last thing I'm asking for you today, and then we can finish this difficult lecture, is what's the next?
The meeting number is low, so it will not lengthen the time. For example, if you lengthen the time of one state in the circadian in the period, it will lead to the length of the period.
Yes, I mean, when we say period elongation, what really happens? Maybe only one step of the cycle takes more time, right? Maybe. Okay, I can stop this thing now. This is getting very difficult. So, this is oscillations, right? These are oscillations. But let's just take only one cycle, this one. And let's remove all the other data. So we just focus on one cycle. And that's the time between this and this is 24, for example. The period is 24 hours. But when you say period elongation, what does it mean? It can be very different. Period elongation could be something like this. That is period elongation. But if instead of starting from this point, you actually bring these two together, you can see here that there is a big delay, right? There is a delay of a few hours between this and this. So you know that this step has been elongated. But then you want to know, okay, what is the time it takes from this point to this point compared to this point to this point? And then when you do that, you may actually find out that actually using DAA or DZNet, it's actually exactly the same. So what we are doing is we are affecting only this phase. Both phases are not affected equally. Only the decreasing phase of per2 oscillation is affected by the AA or the Z. This one is hardly affected at all. So this is one of the things, you're right, that can give us some insight on what is happening. We are inhibiting something that is important for the decrease of per2. Okay, you think about it, and then maybe next week we can sort of discuss. Okay, I think I will let you go now. It's 4 o'clock. I've had enough. If you have questions, don't hesitate. So this is the research I did maybe 5 years ago. It's published, so there is nothing. There is nothing. There is nothing. I forgot the name. Confidential. There is nothing confidential.
Will it be continued to next Thursday?
Maybe not,
No.
because half of the people are not there today. So I'm kind of wondering. Maybe I will make a brief. If we continue, then I will only for 5 minutes briefly explain what we said today, and then pick up from there. So we might continue. We may continue. Do you want to continue? Or is it too difficult?
I want to know the answer.
Me too! I know the answer, but okay, so let's continue. We can continue.
Usual classes is more difficult, I think.
The usual class is more difficult.
Oh.
Yeah, I think because there is more information directly coming from the paper. Yeah, that's true. Then that's nice.